Bruce, are you there?
I am.
Okay. So do you want to go through your presentation and wait till the end for questions, or do you just want people to ask as they come up?
Ask as it comes up. It's more fun that way.
Okay, good.
You're going to have to give me access to my screen.
Oh.
Because I can't share if I.
Yes,
This
hang on.
host disabled participant screen sharing.
Yes, hang on. Let me figure.
I'm not that good to look at, so my PowerPoint is much better than looking at me.
Hang on. Do I make him co-host, Kate? Is that it?
Yeah.
Okay. That will make it easiest.
Yes. Okay, you should be enabled now, Bruce.
So talk to me about, so is this American Grasslands?
This is the Native Prairies Association, and we're the Fort Worth chapter.
Oh my gosh. Well, okay. That's no sweat. I just I thought I don't know, I get them all mixed up, and I don't want to share the same things with people, but some are members of more than one organization. Some of this is going to be repetitive.
Oh, yeah, don't worry about that. A lot of people belong to multiple organizations. That if somebody has seen your presentation before and they decided not to visit it again, then they just didn't register. We have about 50 registrants.
And do they show up like my class 5 minutes after?
Sometimes.
This Zoom call, what would you call it? Not ethic, but
protocol, or?
Well, we've learned that there are certain things that we can do when we're connected virtually, which is you don't have to have your camera on.
True.
Right? You don't even have to have your microphone on. I mean, you can just be listening.
True. And
I find that that's what my students like the most. But, I have to engage them as often as I can to make sure that they haven't, fallen asleep or just sort of, started daydreaming. And in many cases, it takes some minutes to actually respond. And I'm thinking that it's not that they're daydreaming, it's just that they've wandered away and are either washing the dishes or, making their bed or cleaning the kitchen.
Yep.
You never know. And
I'm sure that's all true.
What's that? Yes, but it seems to be okay with everybody. No one's harassing us for, doing it the only way we know how.
So now, do you want your video? Do you want people to be able to see you, or do you just want to be?
I'm distracting. I'll share my right at the beginning so that people can see that I'm not a robot. But then I usually opt to go straight to the presentation, because that way it focuses. I have some odd mannerisms. I tap my finger and nod my head and stuff like that. And those ticks are very distracting for some.
Yeah.
So I just sort of try and stay out of it.
So I don't really have any kind of intro info on you, except that I know you for a long time from Texas Wesleyan. And if you want to add any other history, I know you have a very interesting graduate experiences in Mexico with your corn studies and things like that. So,
well, but listen, you give me credibility. Just knowing you and you inviting me, that's credibility enough, Suzanne. So no more needs to be said.
Okay. Well, yes, but you deserve a lot of I think, a lot of credit for all the schooling and all the research you've done, and the fact that you've picked Tandy Hills as a research location, which is one of my favorite spots. So,
Yeah, well, listen, I was out there just the other day, and man, you know, I remember legumes. I forget the name of them right off the top of my head that used to be there right in, you know, in February, March, and they're no longer there.
oh,
And I think we need to burn the place down.
well, there's been some noise in that direction for quite a while, and every so often some gender reveal party or fireworks go awry and a little patch gets burned, but the Fort Worth Fire Department is quite efficient. They get on it really fast, so not much of it ends up getting burned. But
but do you think we can? Do you think we can organize it? Do you think we could? Are there enough people in the native prairies and master naturalists and?
well, actually, the fact that Britt burned their prairie on, University Drive, practically in downtown, is a good omen.
Yeah. Yeah.
And the Fort Worth Fire Department, they've been doing a lot of in-city burning to practice for wildland fire interface things that might come up. And I think Tandy Hills would be a great place to practice. So that might be way more doable than we would have thought 10 years ago, for sure.
Well, I just think that there's a bank of seeds that is just waiting to be robbed by fire.
Yes.
And, we'll see. I suspect we'll see all sorts of odd things come back. But, I had a student do work on the photinias that are in Tandy.
Oh, yeah? What kind of work?
Well, so what she did is she took the leaves, we dried them, and then she put them in water overnight. And then we took inland sea oat seeds, and we put it into a growth chamber. And she took the stock solution and then divided it by half, and then did a control with distilled water. And the differences in germination rates are, black and white. Photinia inhibits germination almost by an order of 10.
Oh, wow.
Yeah. So, and we were going to actually look at the privets, both the Chinese, the Japanese, and the third one, the glossy, but they all froze, so the leaves we lost the leaves. So we had to sort of resort to something else. But we're gonna probably do that this summer as well. And it really sort of yells at you that, it's those invasive species that are controlling the destiny of the community succession in those prairies. And if we don't get rid of them pretty soon, there's not going to be a lot of chance they'll come back.
Yeah.
And that's my worry,
Yeah.
So.
That's really interesting about Photinia being in the rose family. I've never really heard of anything in the rose family having allelopathic property.
Well, I wasn't too surprised. The Japanese have been doing work with Photinia for a while.
Oh, okay.
And what they were able to do is they use 6-well incubation trays, and what they do is they will put seeds into those with water, but they'll put the leaf just over the top and then cover it so that the green leaf sits in there for a week or so without drying out, and just the volatile compounds from the leaf will inhibit germination.
Wow.
Yeah.
Huh. Well, that's very, very interesting.
Well, so, it makes good sense to plant photinias in your front yard if you don't want anything else to live there, right?
Yeah. Well, it's funny because I had never been quite as worried about Photinia as I was about privet, because it wasn't as, abundant. And I guess I should have been more worried about it.
Well, I think we we give it a lot of space because it looks so good out in our front yard.
Ah.
I've sort of adopted Ugnatia Mexican Buckeye, and it's such a slow grower, but I'm gonna look at that as an allelopathic species.
Oh, really? That's
well,
interesting.
Hoping, it's native.
Yes.
So I'm going to use it on some of the bird seed that I buy. You got sorghum and you got panicum and you got all these weedy species of grass and forbs. And so I was going to take some of those and then see if I couldn't inhibit them with native species, do the reverse. Right?
Yeah.
Just to have fun. I mean, it's, it seems logical. It seems, that Ugnatia is pretty good at holding its own.
Yes.
And what was the other one? Oh, and then elbow plant.
Oh, yeah, I can see that. It's pretty rambunctious.
It is. And, it really withstands the toxicity of the junipers.
Oh, that's true. It makes a skirt around them.
Yeah. So I'm wondering if we might be able to sort of, move along the natives to get rid of the invasives. Then we'll see. I got a couple students interested. So.
Well, gosh, probably 10 or 15 years ago, there was a lot of, I thought, good work and results done using Indian grass and Maximilian sunflower to crowd out Johnson grass. And that seems to have kind of been one and done or something. I haven't really heard about anybody working on something like that again. But
they're doing that, Suzanne?
Well, I think it was Marnie Rout, when she was a master's student at UT Arlington, she did work on Johnson grass and the allelopathic chemicals it produces. And I think anecdotally, she had noticed that Indian grass and Maximilian sunflowers were pretty good at restricting and kind of beating back Johnson grass. And then she went and did her PhD in Montana. So I don't know that she ever really continued that kind of work. And she was back in this area for a while, but I don't know if she's still in this area or not.
What was her name?
Marnie Rout, R-O-U-T.
When I was at the nature center in the West pasture, I noticed that silver bluestem seemed to be fending off Johnson grass. And I asked around and kind of researched it a little bit, but couldn't find much on it. But it seems like there may be some properties with silver bluestem that are able to compete with Johnson grass.
That would be good.
Yeah, so is it allelopathic or is it just competitive?
I don't know. I never thought of silver bluestem as super competitive, but in that pasture, and sometimes when I see it on the roadside with Johnson grass, it seems like it's holding its own. In the West pasture, it was really defined. It was like the whole West pasture practically was Johnson grass. And then there was like this, I forget how large, say 50 by 50, a 50-foot circle or something that was just silver bluestem with no Johnson grass in it.
Well, they're almost congeners, so
no.
I would think I would opt for competition before allelopathy, but allelopathy is one of these factors that we don't, invoke often enough when we're looking at competition.
Yeah. We don't know what's going on in the soil in half the time.
Exactly. Yeah.
Or most of the time.
Yeah. That's so true. It's because it takes a lot of effort. You have to sterilize the soils to be able to run the experiment. And then you've got to find the inoculum, and you've got to be capable of, culturing the inoculum.
Yeah.
And you've got to be able to see whether or not it'll grow on the grass. Yeah, it's not trivial. but it's definitely worthwhile. And seems like Johnson grass just is so prolific. It's found its place here. So it just rained here, and there's more coming.
Yes.
So and I was going to look at my phone to check and see if there's any.
We don't want you to lose connectivity, so.
Yeah, let me check, see if there's something pending.
Well, while you're doing that, it's 7, and I'm just going to make a real short announcement. Next Saturday week, the 22nd, is our next Prairie Seekers training. And unfortunately, all the slots that we're able to offer are full. But we are offering a introduction to Prairie Seekers for anyone who wants to join. It's a Zoom on the 19th at 7 p.m. So if you're interested in just learning more about the Prairie Seekers program and watching it, we're just going to go over the forms and talk about how we set up the whole workday. Then you're welcome to join us. And anyone who's actually registered for the Prairie Seekers class on the 22nd, of course, you're very welcome to join, so you get a little advanced view of what we're going to be talking about on the 22nd. And we hope to be able to offer more of these in the future more often, especially since now we have Dr. Carly Olakey on staff with us in North Texas. And she's a real asset to our organization. And I think she's going to really be able to help us a lot with offering a lot of different kinds of classes and field trips and things. So what do you see, Bruce?
I think I'm in the clear for at least half an hour or so.
Okay, that sounds good.
Well, we'll see, and if not, I'll just say a hasty goodbye, and we'll do whatever we can do, right?
Yeah, well, we had a little marble-sized hail here for about 45 seconds, about an hour ago. So it's spring in Texas.
Yeah.
So welcome to the regular monthly meeting of the Fort Worth chapter of the Native Prairies Association. And I'm Suzanne Tuttle, and I'm the treasurer for the chapter. And Kate Morgan, our president, is here. Hey, Kate. And Michelle, our secretary, and Lorraine, our vice president. So all of our officers are here, and lots of other members and lots of guests from across the state. And we really want to welcome you to our meeting. So our speaker this evening is Dr. Bruce Benz. He's a biology professor at Texas Wesleyan University in Fort Worth. He has a really interesting career and did his graduate work on different varieties of heritage corn in Mexico. So that's a whole nother very interesting topic that I've heard him speak about. But tonight he's going to talk about one of my favorite endemic plants, Silphium albiflorum, the white rosinweed. And Texas Wesleyan University, for those who are not from the Fort Worth area, it's a private college very close to Tandy Hills Natural Area in East Fort Worth. And Bruce has adopted Tandy Hills as one of his close-in research sites for his students. So he's been doing this study on the Silphium albiflorum and their pollination strategies in isolated colonies for quite a while. And I'm going to let Bruce take over. And Bruce would like for y'all to just interject questions anytime you might want to. Don't wait till the end.
Stop me when the urge comes over you. So thank you, Suzanne. And I'm happy to be here. I know most of you, Michelle and I have, and Suzanne have spent hours together at Tandy Hills. Not the last time was 2016 when we ran the Bio Blitz out there. And it's a great place, a remnant prairie. It needs people interested in it. And one of the species that is fairly common in Tandy Hills is white rosinweed. And white rosinweed is I think a fairly typical cross timbers, ecoregion endemic. Here shown with another cross timbers, not endemic, but largely restricted, the scissortail flycatcher. And what I want to
Could
do
I
today
interrupt you just a moment? Suzanne, did you want to record this? I can't push the button because I'm not a co-host.
It's already been recording. I think it's set up to automatically start when the first person arrives. So it's been
Okay,
recording.
Okay, sorry
Yeah, I
to
checked
interrupt
it when
you.
I first got here, so.
Okay,
Okay.
okay, sorry.
So what I want to talk about is a little bit about the species in general, what we know from you know, the general lines of inquiry that exist from citizen science, and then where that line of reasoning took us to actually ask more specific questions about the species and its numerous populations, leading us through the demographic approach, then followed by the genetic approach, and then more recently referring to sort of a community ecology approach that I had not anticipated, except when I first started, that has become sort of a real avenue of intriguing study that doesn't seem to have any end. And at that point, I'll sort of surrender the microphone back to whoever wants to ask questions. So when I started working in Tandy Hills in 1997, I recognized Silphium albiflorum as a relative of Silphium laciniatum, the compass plant. As a grad student at the University of Wisconsin, I spent time collecting for some of the systematic botany courses. And we would visit all the protected areas in southern and middle Wisconsin. And Silphium laciniatum was a common organism. And it merits discussion and interest because it is such a striking plant. And so when I encountered albiflorum after moving to Texas from Mexico, I thought it was at first, until I moved slightly down slope in Tandy Hills from the top up on View Street, going north down to what I call the Trinity River Breaks, and encountering Silphium laciniatum. And so I could see in one place this relationship where they seem to have distinct patterns of occupancy, with the laciniatum growing further down slope, whereas the white rosinweed much more common up on top, where the limestone crops out. As I said, Silphium laciniatum is up here in Madison, Wisconsin, and areas nearby. The genus is well known. It's endemic to the eastern United States. So from an evolutionary point of view, the whole group really is worthy of study, because the various theories about how adaptive radiation and speciation and what we know as the taxon cycle appears to actually be fairly abundantly demonstrated by the various narrow endemics that have evolved out of the wealth of species and geographic opportunities in the eastern United States. So this is a map of the species that were, of the locations for Silphium albiflorum in north central Texas. And we can see here the Metroplex, Highway 35, west and east. So Fort Worth, Dallas, Waco down here, getting down to Troy, and further south down here to Austin. And so this is the aerial extent of the species. And it seems to prefer these light blue dolostone, a carbonate sediment that's greater than 50% by weight, dolomite, or the light green areas, the sedimentary particles of sand and consolidated sedimentary sand, and less so the yellow, the glacial marine sediment that were eroded and derived sediments from marine environment. And the thing about silphium is that if you can see the dolomite, the white limestone, in the aerial view of Google, of iNaturalist, without doubt, you'll find Silphium albiflorum. And it's a very competent plant when it comes to living in these limestone outcrops, suggesting, in fact, that it is able to cope with the relatively high pHs that are associated with the limestone environment. So when I started working on silphium, I talked with Dr. Carey, who is an old colleague of mine from southwestern Colorado, who is a palynologist, a student of the botanists at UT, and she was working for the Texas DOT, and of course, clearance for road building and for power lines and pipelines and the like required an EIS statement that involved looking at the impact on plants and animals. And I asked her about, Silphium albiflorum. I said, it's endemic and it's narrowly endemic. So I suspect that by its very nature, its rarity would suggest that it's probably on the verge of threat if humans are encroaching, or if humans are not encroaching, it is probably a species that is sort of at the end of the taxon cycle and maybe not long for this world. And I was told by Dr. Carey, she said, no, it's not even on our list. The species appears to be, thriving in Texas and doesn't need protection at this point. So I looked at the IUCN criteria for evaluating extinction threat. And this is the basic categorization, right? That not evaluated was where silphium was when I started. And it was probably not evaluated, and we don't care actually when I started. And it was in fact data deficient. We had maps from herbarium records, and there's a couple of herbarium records that are in the global environmental facility that is a clearinghouse for the iNaturalist data and other herbarium records. And it shows that Silphium albiflorum actually is known to occur in the panhandle of Texas as well as in southern Nevada. Well, so I've looked at those specimens. I've looked at pictures of those specimens, and they're not Silphium albiflorum. They're not even probably Asteraceae. In any event, it was obvious that the deficiency of data was enough that it was worthwhile engaging my students. And I have to tell you that my students were not particularly interested at the outset. In some cases, I took them to Tandy Hills or to Stella Row and Prairie, kicking and screaming because they didn't want to spend time outdoors in the middle of September or October in Texas. In any event, what we decided to do, that in order to actually assess extinction threat, we needed to actually embrace the IUCN Red List criteria. We had to look at geography. We had to look at population structure, both age and vital statistics, as well as genetic structure, because it's associated with demography, and then community relations. And that's what I'll talk about in the course of tonight's talk. So the IUCN criteria that is used to quantify the level of threat in a very general way incorporate two measures of geographic distribution: extent of occurrence, the total area the species occupies in square kilometers, and the area of occupancy, the area in which at least one individual is present within a 2 kilometer square area. This is standard procedure for constructing conservation research, and although there's been a lot of discussion about whether or not EOO or AOO or both or some modification of them is appropriate. These are still relatively simple means of assessing threat, and are generally used and embraced by the IUCN and other conservation researchers. So what we did when we started was, and this was after iNaturalist had begun to record evidence for natural biota. And so what we did is we looked at the AOO and the EOO of Silphium. And according to the evidence today, the extent of occurrence, EOO, is large enough that it, in fact, does not have its least concern. But if we look at actual area of occupancy, the amount of area encompassed by the individual populations, it is in fact endangered. And I have to tell you that my students got a kick out of this, because when we started to do this in earnest, there were very few recordings of Silphium, of white rosinweed, in Texas. And so in 2015, it was low concern, but it was endangered. In 2016, as more people recorded this beautiful white aster that is in full bloom on the 4th of July, was still endangered. And as we moved up until the recent, there has been some suggestion that in fact, we may get to a point where the areas here, if they in fact contain Silphium albiflorum, might in fact reduce the vulnerability. Now, I say that, and in the same breath, I want you to realize and recognize that the area that is of the highest density here, and it's recorded in, the Metroplex, so you can expect people are quick to pull out their phones, whereas, down here south of Brownwood or over here in Comanche, it probably isn't as probable that people are going to whip out their phones and take a picture of white rosinweed. So maybe this is an artifact of population and the number of individuals with cameras in the vicinity of the species. And so this is a population in the right of way on Highway 67 north of Midlothian, which is south of Dallas. And many of the populations observed in iNaturalist and collected by herbarium botanists are found in the highway right-of-ways. And some of my colleagues actually poo-poo the evidence as being an artifact of road building. And yet, if you actually look at these populations, there's a population south of Temple, for example, where the individuals are actually coming through the asphalt, and which suggests, in fact, that they were paved over and have come back. So they predate the construction of some of these roads. And the outcrops that are formed by road building are the likely, places where Silphium seems to thrive. So maybe we're, by building roads, actually enhancing its territory. Like so many things in life, the road building has a downside as well, and that downside is that mowing contracts let by counties seem to focus, at least in some parts of the range of Silphium albiflorum, at about the time when the plant is in the midst of flowering. And the result, of course, is that the plant does not produce seed. It is a perennial, it comes back from rootstocks year after year. But we don't see then any sort of maintenance of genetic variability because it's not reproducing sexually. So I said the concern for population viability is fundamental to the IUCN Red List criteria. Population reduction based on direct observation, or the projected inferred based on effects of introduced taxa or parasites. So what we did is we sought out to actually characterize populations in a manner that is commensurate with the way it grows. And so we see here, this is a data recording sheet on which a number of individuals of Silphium have been laid to take a photograph. And what we see is that the plant produces a leaf, about a leaf a year, maybe slightly more, maybe slightly less, depending on the environment. And although this has been only marginally tested, it does seem to suggest that the greater the number of leaves produced, the older the individual. And so even if we were to not actually identify the number of leaves as an indicator of age, perhaps in years, we could use the 2 here, the 4 here, and the 2 here as suggestive of relative age. And so what we did is we use leaf number as a way of describing population structure. Now, we also then, in usually the fall of the year, count the number of seeds produced by an individual by actually pawing through all of the capitula that are produced by every individual plant. And what this allows us to do then is to actually calculate and characterize using what are called static life tables. And so when we see a population like this in the height of growth in the 1st of July, this a collection of individuals near Ponder, Texas, which is in a road that I swear maybe 3 people to 10 people on a weekly basis pass by it. But this ended up on iNaturalist. Anyway, in this population, what we would do in September, October, is to actually paw through each of these individuals, count the number of leaves produced by every rootstock, and then those that had flowers, actually pull the flowers and then count the number of seeds that are actually produced by every individual flower. And we drop the seeds right where we found the plant, so we don't actually remove the genetic potential of the sexual reproducing individuals. Now, the plant defends itself, as the common name suggests, by exuding a very sticky amber-colored resin. And as you handle the plant at any time of the year, you walk away with sticky fingers. And this is one of these parasites, potentially, in a moth that has as yet been not identified. And it is such a mimic for the bracts that actually are found to subtend the capitula on these lateral branches of a flowering individual. So what we did is we actually then looked at survivorship and calculated some of the vital statistics that one can using these demographic data that we collected. Now, this is a comparison of 8 populations that we have characterized over the course of the years. These are all from 2019. And what they do, it's a static life table. So we're looking at log survivorship on this axis, and on the x-axis, we're looking at stage class. And I use stage class in lieu of age class because we don't know for sure that there is a rate of leaf production. And I think the fact is that it's variable enough depending on environment that it is a relative measure at best. But this is typical of what we would call a type 2 survivorship, where the average probability of mortality is equal no matter what the age of the individual. And these locations, so this is the southeasternmost individual, Rutherford Lane. Mason Mountain Wildlife Management Area is the westernmost and southwesternmost population. Tandy Hills is one of the northern, Oak Cliff, about the same, but they're both in the middle of an urban area. Cedar Hill is a nice conservation area. Stella Rowan, likewise, is a conservation area. So many of these areas that we've looked at Silphium's prosperity is in areas that are set aside for conservation-related activities. When we actually then use the static life table, and I'll show you an example in just a minute, we calculate what's called net reproductive value or reproductive value. And reproductive value is the average number of individuals produced through sexually reproducing by an average individual over their lifetime. So it's the total number of offspring that could be produced on average for an individual in that population, or you can look at it by age class or stage class. And what we see here is that to sustain itself, white rosinweed requires a net reproductive value of 1 to be maintaining, to sustain itself. And we see that in some cases, in fact, we've got quite a range of variation. And in other cases, we have no variation at all. In fact, that the populations don't seem to be effectively reproducing themselves at all. That is, the net reproductive value is right at 0. Now, there are some. Mockingbird and Midlothian seems to be doing okay. Oak Cliff is, well, somewhat holding its own. But we get out to Mason Mountain out in the middle of the wilds of Central Texas, or in Cedar Hill, we see, in fact, that the populations are maintaining themselves. Though there is the chance, because we're looking at the 95% confidence interval here, there is a probability that these are also not effectively reproducing themselves, leaving only 2, Mockingbird Nature Park and Stella Rowan Prairie in South Fort Worth, that are actually sustaining their populations. We then look at Rmax, or the intrinsic rate of increase. And this is basically the difference between birth rates, per capita birth rates, and per capita death rates. And so a value of less than 0, it means the population is declining exponentially, and that greater than 0 is going to be increasing exponentially. And if you note here that most of the populations are, in fact, include 0, only Mockingbird and Stella Rowan that are actually effectively reproducing themselves. And this is what a static life table looks like: stage or age classes, number of individuals per age class, total number of seeds produced by individuals of that age class, the reproductive rate, the fecundity schedule, number of seeds per individual, overall individuals, and then survivorship, age class specific survivorship. And then what we do is we sum, that is survivorship times fecundity, and that gives us RO. And then we take that and we multiply it times the age class and divide that by RO, and we get what's called generation time. And what this indicates that we've got at this particular location in Stella Rowan, the average age at which seeds are produced is the 6-leaf stage. And this gives us a feel for sort of how the populations are operating. And it helps to know how the population is structured, because we see here then that the vast majority of the individuals at Stella Rowan are actually not engaged in the reproductive process, right? They're growing and developing. So there's only a few individuals that are actually engaged in the reproductive process to propagate the population in situ. So from that, we then move to genetic estimates of structure. And this was work done by a student of mine who is just in the midst of graduating this week. And what we did is we submitted about 100 samples for genomic analysis. And what they did is we extracted the DNA, they sequenced it, and then the genomic analysis was then sorted in a manner so that we could actually characterize the structure of the populations. And we used a program known as Structure. And what this does is it seeks to see how much genetic variation can in fact be subsumed under certain categories of population similarity. And so we ran this at N of 1, or a K of 1, a K of 2, a K of 3, and K meaning the number of distinct clusters of populations, all the way through K is 10. And the optimal, that is the best solution, ended up showing at N or K equals 4. So of all the populations we looked at, and this is throughout the entire range of the species, it seems like the populations seem to cluster in sort of 4 major groups. And we can see here on the top banner here, we see that there's the Trinity River drainage, and this, as I'll indicate down below in the next one slide as well, that some students of mine back in 2019 at the Texas Plant Conservation Conference at Britt actually hypothesized that in fact the genetic relationships of the species actually could be traced through the major watersheds that the species covers, which is to suggest that actual gene flow or ancestry and descendants actually are confined to particular regions of particular drainages. And that Robbie McManus's analysis actually seems to hold this as true as well. So we've got a number of individuals that are genetically quite distinct from anything else, and they share some of that genetic variation with some of the other populations nearby in the Brazos River drainage, the bluish color. Then the Trinity populations, mostly light blue, all seemingly sharing a great number of allelic variation here. This would be the SNPs, the single nucleotide polymorphisms on the x-axis, and the populations, the individuals themselves on the on the x-axis, and the alleles on the y-axis. So the Trinity seems to hold a very sort of general similarity of alleles that are shared only slightly with the Brazos and the Colorado. And then, so the Brazos is the sort of the most diverse. It contains both the 4 classes, the light blue, the 2 blue, and the 2 green, whereas the Colorado only slightly looks at, holds some of the genetic variation in the blue, and is mostly of the 2 major groups, the light green and the green. And if we look at that then a little bit closer and look at the sort of the sub watersheds, the same seems to hold forth. That is, the groups seem to cluster together, showing like with similarity. And so populations in these stretches of watersheds seem to hang together fairly effectively. Now, Robbie thought that there was some evidence that some of the higher elevations seem to be somewhat distinct from all of the others, but only the high elevation types as well.
So Bruce,
Yeah.
I got a couple of questions.
Shoot.
One is, I read on wildflower.org, which is the Lady Bird Johnson Wildflower Center website, that white rosinweed is long-lived and its taproot reaches 15 feet. Is there a way to age a plant based on the number of leaves? And seed is available at Native American Seed and probably other sources. Is it wise to introduce this species in other locales in which it is known to occur, or where it's diminished in population?
Well, okay. So there's lots of details in there. Yes. So when they built the Chisholm Trail Parkway, some of the local naturalists actually dug out a couple of individuals of Silphium, and the extent of the root penetration was actually greater than 15 feet. It was like, it was 50 feet. It went down and down and down and down and down and down and down. So it has the ability to really persist. And I have thought maybe what we need to do is, get down real deep and then carbon date some of the cortex, see how old it is. Introducing, so I've spent, since I first started to try and germinate these in 2008. I was unsuccessful with getting consistent germination until I actually included limestone in the pots with the seeds. And now I can get germination rates exceeding 85% consistently if I make sure that I've got enough limestone in the soil mix. Now, what that suggests to me is that sort of broadcasting or spreading the seed of Silphium is only going to be effective. That is, it's only going to pick up the opportunity to grow if, in fact, it finds that limestone, that either the basement or it's got limestone on the surface. So I'm not convinced that I bought some of the Native American seed that had Silphium in them, and I spread it on my at my lake house in Possum Kingdom. Now there the sediments are slightly different, and I watched where the Silphium went and sort of stuck flags in the ground to see if it would germinate. And most of the time the seed didn't work its way into the soil well enough so that it actually was capable of germinating. And about half of them were actually removed by local seed eaters, birds and rodents and the like. Now, that shouldn't dissuade anyone from trying to get it to grow anywhere, right? And so I have, like I said, I'm pretty successful now at getting it to germinate, but the conditions have to be right. I don't know if I answered that question completely. I wouldn't dissuade anybody from spreading the seed. The chances of it actually growing, though, are going to be fairly improbable unless you're going to sow them on limestone. Another question, Suzanne?
Yes. If the Silphium are mowed frequently enough, will they put more energy into leaves and then try to quickly send up a flowering stalk?
I've not seen that. I've seen them mowed in south of Abilene every year, around Stephenville and over by Glenrose, where they get out and they mow right away at the end of June when the grass gets a certain height. And once they're mowed, they don't come back and flower, in my experience.
Okay.
That whether they invest more in leaves, I tried to test this hypothesis back in the 2009, 2010, 2011, when we had a real massive drought here. And we did see a slight increase in the number of individuals that were producing leaves. So what we saw was in a relative abundance in the age stage classes that were above 4, 5, 6, 7, and 8. And there was a decline in seed production. So it suggested to me that, in fact, the drought had an impact on growth strategies. I've not seen it in association with mowing, though. Usually when you cut it off by,
Yeah.
middle of June, it's done flowering.
Yeah, it's already put all of its energy into that, and that's all it's going to do.
Till next year, and then sometimes
Yes.
it comes back. But I've seen some of those populations dwindle, and then depending on the conditions or when they mow, if the mowing regime changes, they'll come back. So mowing isn't detrimental necessarily.
Okay. So at the end, when you're finished with your presentation, I have a photograph that one of the members sent to share on my screen for you to take a look at. So,
Yeah.
So anyway, please continue.
Okay, well, more of the same. So the single nucleotide polymorphism data matrix, actually, when you analyze it and sort of reduce the dimensionality, it actually does show that there's nice patterns of the Brazos here in blue, the Colorado in red, and the Trinity River drainage in green, supporting the idea initiated by Alyssa Hutchinson and Ms. Loflingham, that in fact what we're seeing is sort of the populations prosper relative to one another within these watersheds. And likewise, if we look at it even closer, at a slightly different degree of resolution. Populations found in the same sub-watershed of the Brazos, right, and the Trinity, and then the Colorado seem to hang together, suggesting that what gene flow does exist seems to be mostly local. And you know, we still haven't documented effectively who the pollinators of this species is. We've documented a vast number of moths and wasps and bees that seem to hang out and take pollen and or nectar from it, but whether they're effective pollinators or not has yet to be determined. Something else for us to look at in the future. So then the last little bit of this is when I first started looking at Silphium, and I looked at some of the capitula, the flowering heads, or we look at the stem here, and it's riddled with holes. And then many of the flowers actually never mature because they're infested by some sort of a parasite. And so what we did is we looked in the literature, and sure enough, there's a fairly common chalcidoid wasp that appears to parasitize Silphium. But when I collected a bunch of the capitula and some stems and brought them in from the cold and let them sit on the bench top over the months of December, January, February. In March, they hatch. And this actually is a sort of I don't know if it's going to show it or not, but this is a video, and the pupa are actually coming out. So I've had a number of students. It doesn't look like I'm going to be able to show it, but they seem to be parasitizing the disc florets of the white rosinweed capitula. And these are some of the species. And our efforts to identify who's eating who, there's various classes of chalcidoid wasps. There's the parasite, that which probably is Antistrophus sylphi, that seems to be a seed head parasite on not only Silphium laciniatum, but more than likely Silphium albiflorum. And we identify them at this point. I've sent them off, but the people at the Smithsonian have a backload. They have a long list of things that they're supposed to be doing, and I'm sure mine don't get any priority at all. That we see the Anastrophus and then the Eurytomas, and it's suggested that the Eurytoma is actually parasitoid on the Anastrophus, or the Trimeris, which could be in fact a parasitoid on either Eurytoma and or Antistrophus. And as we continued to look and watch them hatch, and I'm not an entomologist, so I don't have any training, and it took me about 6 months to realize that what I need to do is I need to watch it. If it's gonna start to hatch, I need to actually look at the pupa and watch it hatch, as opposed to pulling it out of its puparium, squashing it and extracting its DNA and using barcodes. And I've used both the COX-1, the cyclooxygenase, the electron transport chain, but I've also looked at the internal transcribed spacer and the 18S and the 28S subunits of the ribosomal DNA sequences. And they're not consistent. The different barcoding sequences don't actually identify the same species every time. And so I'm at a little bit of an impasse, but what I'm waiting for is the people at the Smithsonian to give me some identifications so that then I can use that as material for barcoding, but more importantly, to see who is eating who. Because the other class, beside the parasites and the parasitoids, are the inquilins that actually live in the same puparium and just sort of exclude the other one. In any event, The compass plant, Silphium laciniatum, it's a sister species to white rosinweed. And this is where we're headed next. So we've got the genomic samples all set, and we're sending them for sequencing, and we're going to see if in fact we can't find which of the populations or which area in the range of Silphium albiflorum is actually probably ancestral or at least basal. And so the process of evaluation of the ancestry of white rosinweed is in fact in process. So that's sort of a ragtag. I don't have much more. I can talk about seed germination. We've done some experiments. I need to actually identify some of the people who've been involved over the years. And these are ISD and educators and students of my own that have worked on this. And then one who, J. Niall Fisher, who's accompanied me on, I would say probably 75% of my forays into the field. He can identify a limestone outcrop from Google at 10,000 feet, and he can get me there in a minimum of mileage. The rest are students that have worked on this in one form or another. And then most of the support has come from the Texas Higher Education Coordinating Board, the Vivian Redlich Trust from JP Morgan, and then the Sam Taylor Fellowship from the General Board of Higher Education at the Ministry of the United Methodist Church. So with that, I've got a whole bunch of other slides, but it's in slightly disarray. So maybe some of the questions merit actually bringing them out, some maybe not. So you want to show me that picture?
Yes, let me share.
Let me quit sharing here.
Okay. Okay. So this specimen was found on our field trip to Skip Barnett's ranch in far, far, far west Denton County at the Wise County line. And Lisa Stockdike took this photo and was asking if you had any guess of age of this based on the structure.
Yeah, well, okay, so that's a flowering individual.
Yeah.
So it's got to be older than 4 or 5 leaves because we age it relatively, right?
Right.
So the adults are probably 6-leaved, and I'm guessing that that's probably twice that old. Without actually counting the leaves, I probably see between 10 and 15. The largest, the plant with the most leaves on a flowering stalk was 22.
Wow.
And it wasn't that tall. There was one in the Benbrook Spillway area that actually was taller than any I've ever seen, but it only had like 8 leaves. So I would say that this is probably well established. I've probably visited this site. I know where you're talking about, or approximately where you're talking about. I've collected there quite extensively. And, and there are some great populations along the roadside. I don't know, was this on the roadside or in somebody's farm field?
This is in Skip's Prairie. He has a ranch that he manages for conservation as well as livestock. And so he has—
What's his last name? Skip what?
Skip Barnett. So this is on County Line Road off of 380 between Decatur and Denton.
Yeah.
So right in the middle of the Fort Worth Prairie.
Yeah.
Beautiful.
Exactly. No, there's a couple of sites up there that are just replete with white rosinweed, and that and Yucca arkansana, and then the opuntias. Oh, so such a gorgeous prairie. So that's well established.
Yes,
they're doing all right. I didn't show a slide. I've got a slide that shows the growth where the greatest amount of urban growth is taking place, and it seems to be invading the areas of habitation of Silphium in the Trinity River watershed. And it's because, you know, that those limestone cap rocks make a real good, make for a real good view if you can get your house up on top there.
Yes, and very stable foundations too. So I just see that Silphium habitat is being very endangered in general.
Yeah, yeah.
Let me show you another picture to illustrate your, hang on a second, let me bring it up. So let me
Well, interesting question, Leah. You know, I don't know if you captured from the name of one of those Antistrophus sylphi. It appears that that actual species is restricted to the genus Silphium. So if we're interested in, so is that between Azle and Weatherford?
It is.
Isn't that gorgeous?
And this is on FM 730 between Azle and Weatherford. And I've driven that route many times, and I had never seen this Silphium population. And it's because this gets mowed. This is the highway right away, and it gets mowed religiously, except this one year when we got lots and lots of rain in June and they couldn't mow. And this is what there was. And it was fantastic. My favorite colony I think I've ever found.
No, it's gorgeous. That is a gorgeous photo.
Yeah.
Did you put it on iNaturalist?
I did.
Okay. Okay.
So it's out there. Anyway, I just wanted to share that.
No, well, I don't think that you know, that's a good question, whether those parasites are in fact pollinators. There's a lot of bees on Silphium albiflorum, lots and all sizes, from the Great Plains bumble all the way down to these tiny wasps. And I just, how to isolate them so that we can actually identify what it is that gives rise to the pollination that effectively transfers the pollen. I'm open to suggestions. I've had students out there in the middle of July with binoculars, and they're sitting 5 feet from the plant, and they're observing what visits, and they're taking pictures, and they're using their insect nets, and they're collecting things. And we have barcoded and identified probably dozens of species of insects that are involved, taking advantage of the flower, and yet to identify something that effectively pollinates it. Could be the parasitic wasps. You're right.
So some more questions. Alan's asking, is there an easy way to identify a Silphium species in the field when it's not blooming, such as albiflorum versus laciniatum?
Well, so laciniatum, the leaves that are coming out of the ground are going to be, they're going to come out from a rootstock that is fairly broad, whereas Silphium's rootstock is very narrow, a couple of inches at most, whereas the rootstock in laciniatum I've seen gets 4 or 5 inches in diameter. So that's the easiest way that I can identify them. And some of the other indications that the leaves are not ternately compound or ternately divided, which is typical of Silphium albiflorum, is not so common in laciniatum. By that, you got the lobes that are lobed, right? Ternately compound.
Right.
Ternately divided, rather. Yeah. Yeah. So other than that, and Silphium albiflorum seems to create these, and it's not always, but seems to create these And what did that famous ecologist from prairies, Suzanne Tuttle, call them? He called them boneyards in the wintertime. What did you call them?
Fishbone. They look like fish backbones to me.
Well, they do. And you see them scattered over a broad area, whereas Silphium laciniatum seems to be one or two big rosettes of leaves, and that's it.
Yeah.
So they're pretty easy to distinguish. I mean, it takes practice, but it's not difficult.
So you answered Leah's question completely. I was kind of looking for that other picture, so I didn't hear about controlling the parasitic wasp.
Yeah, and I honestly, the citation, the Tooker article is in Biological Conservation, and what they claim is they say, look, if you're interested in conserving biological diversity, the parasitic and parasitoid wasps are just as important about the plants that parasitize. And I agree with that, right? We've got to find a way that, to see whether they both kind of, provide for each other. The population that I discovered the relationship and where I got all those different insects, all the different parasitic wasps, is the population that is spread from Art Cowson Park in Benbrook and extends all the way to the Benbrook Lake spillway. And it's reached the point where they're actually they're not producing any seed at all. But yet the population, the older individuals come back every year. And what I'm doing is I'm monitoring them to see if, in fact, the populations are aging. And obviously, if you're, interested in parasitizing something, you want to kind of hang out where there's a lot of things that you can parasitize. So perhaps as the population ebbs and there aren't a lot of, flowering individuals, that the parasitic wasps are actually going to find somewhere else to hang out, and may leave them long enough so that the plant actually is able to reproduce sexually. And we'll see then this pulse, this surge of young individuals. And we're modeling some of that with the data that we have. But we don't have enough data on the incidence of parasitism at this point, but we're working on it.
So Bruce, if we locate another population of Silphium albiflorum, do you want us to send pictures directly to you, or do you monitor the iNaturalist postings?
And in the IGBIF, yes, I monitor them both. And so at the same time every year, I pull them down and identify the ones that I hadn't seen before. And yes, that's, it's so useful. Nowadays, it's so easy to get that information. Okay. It used to be that I had to go to, Austin and A&M and San Antonio and the Britt Herbarium to actually see what had been collected. But I realized quickly that Silphium albiflorum, because it's such a difficult plant to put between papers and dry, is not something that is regularly collected.
Does anybody else have any questions? You're welcome to unmute. I think you can unmute yourself and ask if you'd like to just ask directly. Okay. Bruce, thank you so much.
My pleasure.
So interesting. Oh, I did have one other question real quick. Kind of going back to Ann's question about sowing seed. If we don't know the origin of the seed, now that we've seen that you see that the genetics of the populations are kind of breaking out into these river drainages, do you think it would be harmful or neutral to plant seed we don't know the genetic origin of?
No, plant it.
Okay.
Spread it around. It desperately needs it, in its native habitat as the area of occupancy becomes less and less. it's not gonna make hay like it should. And I encourage you to if you can get your hands on some seed, to plant it wherever you can get it to grow.
Okay, great.
It's tough to get to grow. Like I said, I've been working on it for years, and it wasn't. I started actually putting seed into the soil between pieces of limestone, and it seemed to be just enough to push it from not at all to I'm going to germinate.
Great.
Yeah. So it takes time. We're getting it.
Yeah, I just wanted to say that I really love how you incorporated your students into that, and the story really of how you've taken students who weren't so excited about a project like that. And I'm sure you've converted them to appreciate prairies and silphium species. But I feel like that's got to be so impactful on your students to be involved in a study like that, that's so narrowly focused on plants that exist nowhere else in the world except this area.
Well, it is in fact the case for considerable proportion of them. Others are just, interested in getting done. But we've, a number of them have published on it, and a number of them, that have moved on to dentistry or to medicine, still are interested. And so they're avid iNaturalist users these days, for example. And I don't think it can get any better than that, They become an active user of something that documents what they see in nature. We're going to see all sorts of great things.
So Alan, is your hand raised? Did you have a question or? Did you have a question? You got your little hand raised icon up. Okay.
I was trying to unmute there, sorry.
Oh, no, that's okay.
I would just calling attention to the question I'd put about being able to identify, it's so easy to identify the Silphium in the field, that the genus, but when it comes to narrowing it down to a species, that was helpful, the information you provided, Bruce. So I appreciate it for sure.
Thank you. No, and it's the other species of Silphium are actually pretty readily identified too. The keys in Diggs et al. Are really good. They really do.
The flora, the North Central Texas flora. Yeah.
Give it a plug because I know I have used that repeatedly.
Yes, mine is all worn out.
Yeah.
So thank you again so much, Bruce. And next month, y'all, we're going to have Donald Beard, who's the superintendent from Caprock Canyon State Park, talking about the Texas State bison herd. And he would actually like to make the presentation live and in person. So we're going to be trying to find a spot where we can meet. Our regular meeting place at Tarrant County College is not open yet. So if we can't, we'll do it on Zoom, but we're going to try to make an attempt to have a live meeting next month. So.
Well, Suzanne, give me a call if you think you can. You can't find a place. I probably can find you a place at Wesleyan.
Oh, wow, Bruce, that would be fantastic.
I don't know how centrally located it is, but it's on the east side, and we've got a couple of fairly large rooms that I think if I reserve, it doesn't cost a nickel.
Okay.
Okay.
Wow, thank you. That's fantastic. I will definitely be in touch.
Yeah, if we want to talk about bison, we got to do it face to face.
Yes.
Okay.
Okay, thanks everybody.
Take care.
Have a good evening.
Get vaccinated.
Bye-bye.
Bye-bye.